Good morning, everyone. So I'm going to make a start. I'm not sure how many more people are going to be joining this session, but we have a 45-minute session, and the way that we're going to run it is that we will have each presentation, which lasts about 5 minutes, and we have 5 of those. And then immediately after each presentation, we'll do the questions for that presentation. If we've got time left at the end and there's any further questions, then we'll take those at the end. But we should have about 5 minutes or just perhaps slightly less for each presentation. So the first presentation that we've got this morning is Shay. Now each of the presenters have introduced both themselves and their presentation in their pre-recorded presentation. So we'll just directly start by going on to the presentation, and then, as I said, we'll have questions afterwards. What I need people to do, please, is write their questions into the chat box, and then we can present those to the speaker afterwards. So, Pauline, if you could start the first presentation, that would be great. Thank you.
Hello, I'm Shay from Sheffield Hallam University. In this presentation, I'm going to tell you about our study on regenerative characteristics of notochordal cells for the treatment of lower back pain. Intervertebral disc degeneration is a major cause of chronic low back pain as the leading cause for disability and is a socio-economic burden for modern societies. Nucleus pulposus cells, which are forming at the center of the disc and are highly hydrated tissue due to their aggregan and collagen type 2 content, lose their water content during the degeneration process. These tissues are derived from the notochord or notochordal cells, and the notochordal cells will be lost during the adulthood in human. And that's where the degeneration process will start. In some animals that they keep the notochordal cells till the longer age, the degeneration process will start later. Therefore, we thought that the co-culture of nucleus pulposus cells with DMP cells may have regenerative properties on DMP tissue. However, first we need to optimize the culture of notochordal cells in vitro, avoiding the use of bovine serum and using available culture system as the first step in establishing this process, and further we can use the regenerative properties for treatment of disc degeneration. We use the porcine notochordal cells and culture these cells in either 3D or 2D culture. We use alginate beads as a gold standard model of 3D culture, and in this presentation I'm going to focus only on the 3D culture. We culture the beads in low glucose DMEM or alpha MEM, and also in 2 different oxygen concentration, 5% and 21%. Therefore, we will have 4 different culture conditions. And we harvest the beads after 24 hours, 1 week and 2 weeks. The outcome measurements of the culture conditions will be analyzes the NC marker at gene and protein level and some morphology studies as well. For the marker of NC cells, we aim for brochure, aggregan, collagen type 2, keratin 8, keratin 18, keratin 19, and FOXA2, and also collagen 1 as a negative marker of notochordal cells. So the results for the notochordal marker gene expression analysis, I show you the results of brochure, COL1A, FOXA2, and keratin 18, and the rest are ongoing. Brochure relative gene expression did not show any significant difference between the 4 different culture conditions. Same is observed for collagen type 1. We didn't see any significant difference between the 4 different culture conditions. FOXA2 relative gene expression and also keratin 18 relative gene expression didn't show any significant difference between the 4 different culture conditions. We need to have the result for other gene expression and also protein and morphology studies to wrap up this experiment to conclude that and we can continue for local leucostemia for our co-culture studies. Therefore, we can say that optimizing the culture of notochordal cells in vitro could not only help us to establish the culture for further experiments, but also to efficiently expand them in larger scale for therapeutic purposes and treatment of disc degeneration and low back pain. And protein studies and morphology analyses are still ongoing. At the end, I would like to thank all my colleagues in our group, especially Christine, for her guide during these experiments. Thank you.
Can I just ask if anybody wants to ask a question? Can they put it in the chat and I'll bring it to the attention of the speaker? Thank you.
So Shay,
Claire.
Thank you. We have a question here from Janet, and Janet's question, as I'm sure you can read there, is what will the clinical application be in the future, or how do you see it rolling out in practice?
Thank you for your question, Janet. Actually, as I mentioned in my aim, we are going to optimize this culture condition to use the notochordal cells for cell therapy treatments and the treatments of disc degeneration. So we aim to co-culture the notochordal cell. In our project, we are going to use the stem cells that has been reprogrammed to notochordal cells. And we co-culture these cells with degenerated NP cells of the tissue to actually propose some regeneration properties in the NP cells. And with the use of the biomaterials, hopefully we're going to put them back into the degenerated disc and reverse the process of degeneration and treat the lower back pain.
Thank you, Shay. That's great. Does anybody else have any other questions? I can see a couple of people perhaps typing. No. And Shay, is there anything else that you wanted to add? Anything you want to highlight?
There's just one new question come through from Jude. Shaya, there's a question from Jude Meakin. How sensitive are the cells to their environment? Shaya, did you hear the question?
Mute. Yeah, sorry. The notochordal cells are normally cultured in the alpha MEM media in the lab, and since we want to culture them with the MP cells as a co-culture, we need to optimize the culture condition and also optimize the components of the cultures to make sure that they would survive in the co-culture condition, if I have understood the question properly.
I think there's just one more question coming through that we'll take, and
I think that might be it. So should we move on to the next presentation, please? Thank you.
Thank you.
Hello, I'm Joseph Snooks, a postdoctoral research associate at Sheffield Hallam University. Here I'm presenting a Caprine model of intervertebral disc degeneration, a testing platform for an injectable hydrogel. There are many different animal models of intervertebral disc biology and degeneration, yet not all accurately represent human degeneration and have practical, ethical, and biological limitations. Here we aim to develop an ex vivo organ culture model that mimics a moderate level of human intervertebral disc degeneration using discs obtained from goat spines collected from the abattoir. To test the efficacy of emerging treatment strategies such as MP gel. Developed at Sheffield Hallam University, MP gel is an injectable hydrogel showing promising anti-degenerative properties that we wish to investigate further within the intervertebral disc. The model was produced by using a loaded disc culture system in Figure 2 that allows the culture of intact intervertebral discs under physiological loading and media perfusion. Figure 3 shows that Caprine IVDs were preloaded for 3 days and then injected with a combination of collagenase and chondroitinase, two enzymes involved in the degeneration of the intervertebral disc, and subjected to 7 days of simulated physiological loading. To test MP gel, this regime was repeated, and MP gel alone, or with mesenchymal stem cells added, was injected, and discs were cultured for a further 21 days. Moving on to results of establishing the model. Figure 4, during culture and the loading regime, the height of control and sham discs remained constant, whereas enzyme-injected discs in red displayed a reduction in disc height over time. Histological staining in Figure 5 shows the presence of fissures and matrix loss in enzyme-injected intervertebral discs. And investigating protein expression in Figure 6 highlights a decrease in matrix proteins and an increase in catabolic protein expression in enzyme-injected discs, which is all indicative of a moderate level of degeneration in humans. After establishing our model, we moved on to NP gel injection. Figure 7 displays macroscopic images of Caprine IVDs where injected NP gel is clearly visible in NP regions and appears shiny within the red boxes. Below in Figure 8, histological staining shows the integration of and cell migration into NP gel within discs, whereas fissures are still present in degenerative controls. Immunohistochemistry in Figure 9 shows an increase in matrix expression and a decrease in catabolic protein expression in discs injected with MP gel compared to degenerate controls, reversing degenerative processes observed in our model to begin with. In conclusion, we have established a relatively quick, reproducible ex vivo Caprine model mimicking human degeneration. Injection of MP gel attenuated the catabolic phenotype of our model, suggesting it has the potential to halt degenerative processes. Finally, this model may have the capacity to enable rapid screening of novel strategies to combat intervertebral disc degeneration in the future. Thank you very much.
Thank you. For this presentation, Professor Christine Lemaitre will be answering any questions. So if you have any questions, can you pop them in the chat?
Yeah, just to explain, Joseph's in the lab today. We don't have many lab days access, so he's opted to go in the lab.
There's a question coming through in a minute, Christine. Just
Okay.
It takes a few seconds for the typing. So it's a question from Andrew Dixon. How long were the NP gels left in place? Question would the enzymes used in the degenerative process potentially affect the outcomes?
Yeah, so thanks very much for the question there, Andrew. So the enzymes which are utilised have a fairly short half-life. So once they've been injected, we allow sort of the degeneration to take place over a course of 7 days, first of all. And then following that degeneration, there won't be any of those enzymes left. But what's happened during that period of time is the catabolic degeneration cascade that occurs during human degeneration has been triggered by those cells. We then inject in the hydrogels, and those were left for a further 3 weeks. So 1 week induction of degeneration, and then 3 weeks following treatment. And as you could have seen in the images, we see really impressive regeneration of those degenerate discs when we've added the MP gel into those discs.
Thanks, Christine. There's one more question here. I think we'll just take this one more question. This is from Jude Meakin. I may have missed it, but is the caprine model advantageous over other models such as ovine or bovine?
So we're also working on a bovine model at the moment in collaboration.
We unfortunately seem to have lost Christine's audio there, so hopefully she'll be able to come back and continue to answer that question. What I suggest we do is to move on to the next presentation. Thank you.
Good morning, everyone, and welcome to my presentation on in vitro human enthesitis model with induced IL-17A and TNF-alpha from CD4 and CD8 lymphocytes, with the effects of pharmacological antagonism with Janus kinase and retinoic acid receptor-related orphan receptor gamma inhibition. My name is Hannah Rowe, and I'm a recipient of the Novartis UK Investigator Initiating Non-Clinical Research Funding Support. So my work is looking at enthesitis, which is inflammation at points where tendon, ligament, and joint capsules are inserted into the bone, which is a primary cause of inflammation in ankylosing spondylitis. It's been shown in healthy controls that there is a population of innate resident lymphocytes that produce IL-17A, which is a pro-inflammatory cytokine. Specific MHC class 1 association is a large risk factor for ankylosing spondylitis, which points towards CD8 T cells and pathogenesis. However, there's also been MHC class 2 associations, which points towards CD4 T cells. Here's a diagram of disease progression, starting at inflammation, eventually leading to syndesmophyte formation, and then complete spinal fusion known as bamboo spine. So sample procurement, we get these samples from decompression surgeries or scoliosis, where we select parenthesial bone, endothelial soft tissue, and we also get matched peripheral blood from these patients as well. So following on from sample collection, we then undergo mechanical digest. This then goes on to magnetic separation using CD4 and CD8 cells. And then we go through a 48-hour stimulation process in standard culture conditions for ELISA, and then subsequent gene expression in RT-PCR. So we first of all wanted to confirm whether CD4 and CD8 T cells from the enthesis could create SPA-related cytokines, namely TNF-alpha and IL-17A. And we can see in these graphs that the stimulation does indeed induce at both IL-17A and TNF in the CD4 populations with significance, and in the CD8 TNF-alpha population also with significance. And these are just flow plots demonstrating the upregulation following stimulation. So we then wanted to see if we could still have this production of inducible IL-17A and TNF following therapeutic agent application. So this is looking at, we use Rho gamma T inhibitor, which is an inhibitor for the master transcription factor of Th17 cells. We then use methotrexate, which is widely used in rheumatoid arthritis and psoriatic arthritis. And we also use the PDE4 inhibitor, which elevates cyclic AMP and modulates NF-κB signaling. So the first one looking at IL-17A ELISA, and we can see clearly the complete abolition of IL-17A production following raw gamma T inhibited treated cells, which makes sense given the amount of IL-17A those Th17 cells are known to produce. We also see significance in both of these, in both the parenchymal bone and the matched peripheral blood. And we see significance in PD4 inhibited treated cells in both of these populations as well, alongside methotrexate in the peripheral blood CD4 cells. So comparative to the previous IL-17A ELISA I just showed you, we can see that raw gamma T inhibitor doesn't seem to affect TNF alpha production. However, PD4 inhibitor still seems to work at inhibiting this production. So we can see that inhibition in CD4 parenchymal bone. Same again in the matched peripheral blood. No significance in the CD8s, but there is significance and downregulation in that TNF alpha production in PD4 inhibitor treated cells. So moving on from that, we then ran a few RT-PCR experiments ranging from N of 6 to an N of 20, going up like this. And we can see, sort of matching our ELISA data, the inhibition of IL-17A production in RT-PCR following raw gamma T inhibitor treatment. We can then see an upregulation in IL-6 production, again in PD4 inhibitor treated cells. RRC, which is the gene that encodes the raw gamma T transcription factor, that has again an upregulation. And the last two sort of immunomodulatory factors of FOXP3, which is the mass transcription factor for Tregs, and TGF-beta. We can see significance in PD4 inhibitor treated cells. Now, comparative to the last graphs that I just showed you, the CD8 PBMCs didn't show any significance, but we can see that the actual transcript expression is much higher in these cells, which may support a stance of it being more immunomodulatory within this specific CD8 PBMC population. We then looked at a heat map looking at growth factors, cytokines, and I'll just point you through a few now. So TGF-beta, as we can see, is upregulated in blood, parenchymal bone, and soft tissue, and all these are matched. We can then see a downregulation in TBET, which is associated with TRM and TH1. And we also see a upregulation in NR4A1 in both parenchymal bone and soft tissue. I'd like to thank everyone. Just to conclude, normal human entities just have CD4 and CD8 T cells. This results in an inflammatory Th17-like phenotype and a more immunomodulatory CD8 PBMC T cells. Raw gamma T effectively inhibits the IL-17A production following the ELISA and QRT-PCRs. And as I mentioned, methotrexate is widely used in psoriatic arthritis. However, results suggest it may not be as effective in axial disease, highlighting potential difference in disease mechanisms within the spondyloarthropathies. I'd like to thank my supervisors, surgeons, and everybody else on my team for support with this project.
Thank you, Hannah. There's a question from Christine Lemaitre. Hannah, thank you for entering the presentation. How many CD4 positive cells can you harvest from the entheses? Do you expand your cells prior to investigation or utilise immediately following extraction?
Hi, thanks very much for that question. Can you hear me okay? I'm just checking everything's okay.
Yes, Hank.
Yeah, that's fine. Yeah, so we use our cells straight after harvest. We don't expand them. Basically just due to viability. They seem to work more effectively when we use them straight after extraction. But depending on the type of patient that's undergone surgery can massively affect the amount of cells that we can extract from these. So this can range from 1 million cells to up to 10 million, depending on the size of the actual spinal tissue sample that we collect from the surgery. Yeah.
I think there might be another question, Hannah. Just
okay.
See somebody typing.
Mm-hmm.
There's a question from Janet Dean, Hannah. What is the potential structural impact of your research, and how will this impact patients?
So a lot of my work, as you've seen, is looking at the T-cell biology specifically. So a lot of my focus is looking at the sort of plasticity between different T-cell populations. So in theory, once we can sort of identify this sort of T-cell biology at the enthesis, we're hoping that we can target specific cell types to sort of reduce that inflammation, and then obviously hope to improve patients in terms of the ankylosing spondylitis.
Okay. Thank you, Hannah. I think we'll move on to the next presentation. Thank you.
Good afternoon, everyone. My name is Luc van Dijk, and today I'm going to present to you the results of our in vitro study in which we evaluated the response of human macrophages to calcium phosphates with specific surface topographies. Synthetic calcium phosphate bone grafts are commonly used in bone surgery as a substitute for autograft. Preclinical studies have demonstrated an enhanced performance of calcium phosphates that have a submicron topography when compared to conventional calcium phosphates. The enhanced performance of these materials may be explained by a favorable osteoimmune response to the surface topography. Specifically, this means an upregulation of anti-inflammatory pro-healing N2 macrophages, which could explain the enhanced bone healing that is observed with these materials. For the methods, we used 2 calcium phosphate bone grafts for evaluation in this study. The first was a BCP with a submicron surface topography, the second a TCP with a conventional topography. The surfaces of these 2 bone grafts are shown in the center of this slide on scanning electron microscopy images. We isolated CD14 positive monocytes from the buffy coats of 5 human donors and seeded these cells on the 2 bone grafts in vitro. After 24 and 72 hours, we performed various assessment methods, including evaluation of macrophage morphology by SEM, and also we determined expression of macrophage markers specific for the M1 phenotype and the M2 phenotype, both by ELISA and by qPCR. Lastly, we used conditioned medium from macrophages cultured on these 2 bone grafts in an angiogenic tube formation assay and an osteogenic differentiation assay of mesenchymal stem cells. On to the results. First, the assessment was scanning electron microscopy, and actually we determined that macrophages responded differently to the different bone graft surfaces. So what we observed was that on the calcium phosphate bone graft with a submicron surface topography, the macrophages adopted an elongated shape, while the macrophages on the conventional calcium phosphate surface remained spherical. By ELISA, we determined an overall higher expression of M2 macrophage markers on the material with the submicron topography. So CCL18 and CD163 were significantly higher expressed on the submicron topography compared to the conventional topography. For the M1 markers, interleukin-6 was secreted to a higher level at the submicron topography after 24 hours, but this effect had abated after 72 hours, and no differences were determined in CCL5 levels. By PCR, the results were a little bit less clear because we found a higher expression of both M1 and M2 genes on the submicron surface topography. So for example, an M2 marker CD206 was secreted to a higher level on submicron topography after 72 hours, but on the other hand, M1 markers CCL2 and IL-1 beta were secreted to a higher level on the submicron topography after 24 hours. However, after 72 hours, those differences had abated for the M1 markers. Results from the angiogenic tube formation assay indicated that medium conditioned by macrophages cultured on the calcium phosphate with a submicron topography induced more extensive angiogenic tube networks in vitro, and this was evident from the microscopy images as well as the tube network quantification performed by software, in which we determined greater tube length and a higher number of junctions in the submicron topography condition. In addition, in the osteogenic differentiation assay, we determined that macrophages cultured on the BCP with a submicron topography induced stronger osteogenic differentiation of the MSCs. This was evaluated by measuring the ALP activity in MSCs that were cultured for 10 days in the conditioned medium of the macrophages. In conclusion, this study revealed a stronger activation of macrophages on calcium phosphate with a submicron topography, which was in favor of the M2 macrophage response. We determined upregulation of M2 macrophage markers on these materials, and although some M1 markers were higher on the submicron topography at 24 hours, this effect had abated after 72 hours. The enhanced angiogenesis and osteogenesis outcomes in the in vitro conditioned medium assays also indicate a pro-regenerative effect of the macrophages on the submicron topography, which corresponds to the function of M2 macrophages in vivo. Lastly, the elongated shape of the macrophages on the submicron topography also corresponds to the shape of M2 macrophages that is reported in literature. So overall, these results suggest a favorable immune response against calcium phosphate with the submicron topography, and this could be an explanation for their enhanced performance that is observed in vivo when compared to conventional calcium phosphates. Thanks a lot for your time, and I am very happy to answer any of your questions. Thank you.
As you'll see from the note from Pauline, Lucas isn't here today. So if there are any questions, if you pop them in the chat, we'll make a note of them and forward them on to Lucas, and hopefully he'll be able to get back to you. So thank you for that. So we move on to the final presentation. Thank you.
Welcome everyone. My name is Mustafa Yasin. I'm orthopedic registrar, and this is my lecture for the British Spine Conference 2021. It's about amyloid deposits in the ligamentum flavum related to lumbar disc degeneration and spinal canal stenosis. My presentation scheme, I will talk briefly about spinal stenosis, amyloidosis, materials, methods, and results of my study. The spinal canal stenosis, the degenerative type, is a progressive disorder affecting the whole spinal motion segment, where there will be progressive narrowing of the spinal canal, compressing of the motor and sensory nerve, causing debilitating symptoms for the patient, usually affecting the patient more than 65 years old. The calcification and the hypertrophy of the ligamentum flavum is blamed for that. The degenerative process itself is not yet well understood. However, the accumulation of certain materials was blamed for that, like the amyloidosis. The amyloidosis is accumulation of extra or intracellular pathogenic amyloid deposits. The amyloid itself is aggregation of misfolded proteins. We can diagnose this accumulation by certain stains. For example, the amorphous isogenic appearance on hematoxylin and iosine staining, as is shown in the pink slide, and the apple green bifurcations on Congo red histological staining, as is shown in the green slide. Many studies handle the relation between the amyloid and certain diseases like Takanow in 2009. He demonstrated a relationship between amyloid deposits within the synovial membrane and the development of carpal tunnel syndrome and spondyloarthropathies in patients undergoing continuous dialysis. While Yanagisawa et al. In 2014, he studied 95 ligamentum flavum taken from patients with spinal canal stenosis and lumbar disc degeneration, and found that 45% of the patients with spinal canal stenosis tested positive for amyloid deposition. My study aimed to find a relation between the amyloid deposit within the ligamentum flavum to the spinal canal stenosis and or lumbar disc degeneration. It's an analytic cross-sectional study. The consent was taken from the patient undergoing surgery already, and the tissue taken intraoperatively from the ligamentum flavum at the affected level. We took the tissue and fixed it in normal saline with paraffin. Then we sectioned it to certain thickness, stained it with immunohistochemical stain and hematoxylin and iosine stain. The extent amyloid deposition determined by measuring degree of amyloid positive area and categorized into positive and negative. We had many exclusion criteria in order not to affect our results, for example, infection, trauma, tumor. These two slides, on the left, the ligamentum flavum stained with hematoxylin and iosine stain, while on the right, our control slide, the kidney tissue stained with immunohistochemical stain. These two slides showing the positive and negative slides. On the left, the arrow mentioned the brown area, which is the accumulation of the amyloid detected by the immunohistochemical stain, while on the right is black, which means that it's negative. We used the MRI in order to measure the canal stenosis and the ligamentum flavum at the level of the facet joint. This was done by 2 MSK radiologists, and we took the mean. We started with 16 patients with lumbar spine canal stenosis and 11 patients with lumbar disc prolapse. Few of the slides were corrupted during the processing. However, we ended with 11 specimens for both the spinal canal stenosis and lumbar disc prolapse. As we can see the results on this table, we had 5 slides positive out of 11 in the stenotic group, while none in the disc degeneration group. 4 females and 1 male of the stenotic group. The affected level were L3, L4, and L4, L5. L5S1 and L2, L3, we didn't find any positive result in it. The age affected with the positive results were more than 50 years old, while less than 50, we didn't have any positive results. We tried to find a correlation between the mean ligamentum flavum thickness and the result of the immunohistochemical stain. This was positive, as we can see the p-value is 0.001, which is statistically significant. And we tried to find this correlation with the canal diameter, but we didn't get a statistical significant p-value. The conclusion of the study is that amyloid precipitate is a statistically significant effect on the thickness of the ligamentum flavum. Lumbar disc degeneration does not appear to be related to amyloid deposition. Our take-home message is amyloid may contribute to ligamentum flavum hypertrophy and subsequent lumbar canal stenosis, and this is ongoing research area of interest. Thank you. Any questions?
Okay, so as Pauline said, unfortunately Mustafa's not available today. So if you can type any questions in the chat, and then we'll push them on to Mustafa, and hopefully he'll be able to get back to you. Christine, are you still there? Because I've just got one additional question about Joseph's work, if you're available. Christine, can you hear me?
Yep, I'm just connecting.
Okay.
Hiya, can you hear me?
Yeah, I can hear you. There was just an earlier question from Janet about Joseph's presentation. Mm-hmm. Could you comment a little more on the method? Is this a unique scientific method, and what is the potential future impact of the work as you see it?
Yeah, so this utilises, so the actual degeneration model utilises an organ culture system where you can culture whole intervertebral discs under loads that mimic the natural loading patterns that you see during human loading. So this enables us to actually look at potential regenerative therapies in an environment that mimics much more closely the environment of the human disc. The hydrogel system that we discussed, I'll talk a lot more about on my plenary session on Friday. So if you're interested, come and talk to me there. But basically, this hydrogel is one that we've developed. It's a low viscosity injectable hydrogel that has great potential for regeneration. So we were utilizing this organ culture system to test the hydrogel as a potential therapy for regeneration. And the impact could be massive. So it's got really good potential for regenerating the intervertebral disc. So for those some 40% of patients with chronic back pain, which are caused by disc degeneration, we could actually potentially have a new therapy for those patients. Hopefully you could hear me all the way through that time. Can you still hear me? Yep.
Yeah, that was fine. Thank you, Christine.
No problem.
Let me just check if there are any more. There are no more questions. Janet just sent her. Thanks, Christine.
No problem.
So Claire, do you want to wrap up the session?
Thank you, and thank you to all the presenters and also for all the attendees. So unless anyone's got any final questions, we'll end the session there. And then, as I understand it, that will take us all back to the auditorium. So thank you for your time.